Hello, everyone. Welcome to the Science Café presented by UMass Chen Postdoc Association. I'm Ruchi Sharma. I'm a postdoc researcher at UMass Medical School and also part of UMass Chen Postdoc Association in Worcester. This is the outreach event for specifically designed for community members and general public. So thank you, everyone, for joining. Uh, in today's Science Cafe, we'll be talking about unlocking the secrets of brain growth and decline, how glial cells shape our minds. So we have two wonderful speakers, Travis Faust and Patrick Sheehan. They'll be talking about more about how the brain cells connect, develop, change, and what happened when they go wrong. So I would like to tell everyone that this event will be recorded, and you can find this event and various other Science Cafe on Oompa YouTube channel. And you can share it with your colleagues, students, or any family member who is interested in scientific topics like this. We are also active on different social platforms like Facebook, Instagram, Twitter, and YouTube. You can follow us on that for further notification and updates. You can also email us on oompa@umassmad.edu. Just a minute. Yep. So before introducing our speakers, I would like to state that the views presented in this presentation and slides are solely belong to the presenter and not to the UMass Chan Medical School. I would like to also request to the people who will asking question to keep their language simple and accessible for the participant who doesn't have the scientific background, and so that they can follow along and engage into the discussion. Please. Also, meanwhile, during the talk, if you have any questions, please type in the chat box, and my colleagues, Olesia and Oscar, will help to get your questions. At the very end, we will post you a survey link. It will be helpful for getting your feedback. With that, I would like to introduce our first speaker, Travis Faust. So Travis is an instructor in Dr. Dorothy Sheffer's lab in neurobiology department at UMass Chan Medical School. He studies how glial cells, the non-neuronal cells in the brain, communicate with each other to coordinate the fine-tuning of developing brain circuits. He is originally from Wellesley, Massachusetts. He did his PhD in neuroscience at Johns Hopkins in Dr. Akira Shava's lab, where he studied how glial cells contribute to development of mental illness such as schizophrenia and depression. Outside of lab, Travis is an avid sport fan and board gamer, and also father of two young boys. So Travis, with that, I would like to thank you for joining this event, and you can take away from here.
Great. Well, thank you for that kind introduction. Excited for everyone here. Thank you for coming. I'm excited to talk a bit about how glial cells shape the brain and sort of our work at UMass, looking into newer mechanisms of how this works. So let's see. So probably you've all seen a picture of the brain. You're probably familiar with how it looks. It kind of looks like this pinkish blob with all these wrinkles on it. But actually, if you take a little closer look, there's a lot of complexity to it once you start to look at sort of a microscopic level. And if you zoom in on just a piece of the brain that's only the size of a grain of salt, what you can see are that there's actually these tiny little things called cells in the brain. And these ones in the brain are called neurons. And neurons have this interesting structure. They look kind of like a tree where they have all of these different branches. And so does anyone want to try and take a guess as to how many neurons there are in the human brain? You can sort of take an opportunity to put us in the poll, in the chat, and just sort of give an estimate of what's your best guess for how many neurons you think there are in the brain?
Can everybody see the poll? I just lost it now. Yep. Okay, good. Thank you.
All right. I think most everybody's given a guess at this point, and I think there's sort of consensus emerging around 100 billion. And this is, this is the number of neurons there are in the human brain. It's actually closer to about 86 billion plus or minus 8 billion. For a sense of scale, this is the number of human beings that have ever lived on planet Earth is about the same number of neurons that you have in one human brain. And nobody guessed this little, but 15,000 neurons, this is actually the number of neurons that you have in this salt grain sized piece of brain tissue. And so neurons have this structure where there's sort of two different ends. There's one side that receives input from all sorts of different places and then makes the decision whether or not to communicate an electrical signal. This travels down to the other end of the neuron where it has sort of the output end. And you can sort of make an analogy to a thermostat where you're collecting temperature from different rooms of the house. And then you ultimately make a decision whether or not to turn on the furnace and then send heat through the ducts to then heat the rest of your house. And when neurons are connected to each other, we would call this a neural circuit. And the point at which they connect, this is called a synapse. And so if you look over here on the right at this picture of a neuron from a human brain, all of these tiny little orange and blue dots, these are synapses. And there are about 10,000 synapses per neuron, which means in this 1 cubic millimeter of brain tissue, there are 150 million synapses. And in the entire human brain, there are 1 quadrillion synapses. And so what this looks like, there's actually a little bit of a gap between the two neurons when there is a synapse. And so the electrical signal comes down, it reaches the end of one neuron. It then causes these, what we call vesicles, to come to this end of the cell and release chemicals that travel to the next neuron. And that can elicit an electrical signal that will then propagate through the circuit. And given the large number of synapses, it takes a long time to actually form all of them and make sure that the right ones are formed in your brain. And so when a human is born, this is what your neurons look like. And over the course of the first couple of months of life, they become more and more complex. And by about 2 years of age and a toddler, there are much more complex and highly ramified, and there are more connections between them. But actually what happens as you get past that toddler age, you actually start to remove some of these connections. And this is a process we call pruning. This is sort of similar to a tree where you remove the unnecessary branches to make the circuit more specific. And so what this means is that over the course of life, you have sort of initial peak, sort of in the toddler age, of the number of synapses you have in the brain, and then you sort of prune away the excess ones. And this happens in between a balance of two different things, given you have these different synapses. Here I'm showing the pre side, which is one neuron, and the post side, which is the partner neuron. You can have formation of new synapses, and you can have elimination of old synapses. And it's the balance between these two processes that gives you the total number of synapses you have in the brain. And interestingly, the process of pruning kind of happens at specific periods of life. There's one early life where there's a lot of pruning, and there's a second sort of big wave of pruning that happens in adolescence. And if you compare the brain of someone who's neurotypical to someone with autism spectrum disorder or schizophrenia, these different types of mental illnesses, you actually see differences in the number of synapses these people have that emerge at these specific periods of time, either in early life or starting in adolescence. And so we're really interested in the question of how does the brain determine which synapses to remove and which ones to spare? Neurons aren't the only cells in the brain. They're actually supported and maintained by a bunch of other cells, which are not neurons, and they are collectively called glial cells. And so in this whole grain of salt size piece of brain, where you have 15,000 neurons, you actually have 20,000 of what we call oligodendrocytes. These wrap the parts of the neuron that send the electrical signals and make the signals go much faster. You also have 5,000 of these bushy cells, which we call astrocytes. They look kind of like stars. And these ones have the tips of the cell that go right next to the synapse. And then we have about 2,000 of these cells called microglia. And these are the cells which are the main focus of our lab at UMass. These are the immune cells that live in the brain. And so they rapidly respond to bacteria, viruses, damage in the brain. They initiate inflammation, and they clear away cell debris. And so here's a picture of a microglia. They also, living in the brain, even though they are immune cells, they also have a lot of important roles for interacting with synapses. And so one thing they do is they remove away the less active synapses. So if you have two synapses here with their levels of electrical activity, and you have one that gets more activity with more of these spikes, or one in which the electrical activity decreases and it sort of flatlines, the microglia will come in and it will remove the less active synapses. But we're still sort of working out all of the different ways that a microglia knows to come in and remove the less active synapses. And one way that we can study this is taking advantage of the fact that there are specific areas of the brain that are activated by different senses. And so there's one part of the brain in which responds to when you get touched. And within that, there are specific parts of that that respond to touch to every different part of your body. And so if you were to sort of lose touch to your thumb or have less touching on your thumb, then you expect the neural activity in that part of the brain to be reduced. And so we use mice to study this. Mice receive most of their sense of touch through their whiskers. And so there's a part of their brain in the same area which has different sort of looking like circles here for every single one of their whiskers. And so each of these would respond to activity and touching of one of those whiskers. And what we do is we take the mice and we remove the whiskers on one side so that they no longer are able to sense and be, that sense of touch. And so what we see here are all of the synapses in these parts of the brain that are activated by touch to individual whiskers. And we compare one side of the mouse to the other, which still has the intact whiskers. And what we find is that here we're showing the groups of synapses in this part of the brain. After a few days, you start to see this loss of these synapses because we've reduced the amount of neural activity going to those that part of the brain. And what we've now come to understand about this process is that when we do this, the microglia are able to sense which synapses have a reduction in activity, and they then interact with the astrocytes, which I mentioned are these cells, the star-like shells, which are normally right next to the synapses. But what happens is that the astrocytes move away from the synapse, and this is important because it allows the microglia to come in and remove the synapses. And what's interesting is that if you prevent the astrocytes from moving away, the microglia are not able to come in and remove the synapses. And so the microglia have a way of signaling to the astrocytes that tells them to move away from the synapses. And this is a really important part of how these two cells coordinate with each other to remove synapses in the brain. And so I just want to thank everybody from the lab, sort of in the Schaefer lab. These are all the people who work together with me. These are all people here at UMass Chan who worked on this, as well as people from other institutions who have helped us on this research. and I'm sort of happy to open it up and take questions from the group about this or anything I've talked about here.
Great. Thank you, Travis. I would like to encourage everyone, if you have questions, to please type them in the chat. And maybe I will get started by asking the first question. So I had a question about the microglia and their dual role in fighting infection and also in pruning the synapses. So my question is, are there different types of microglia that do these functions in the brain, or is it the same kinds of microglia that do this and they just change the way they function depending on the circumstance? Let's say in a developing baby who also happens to get an infection in their brain, do these same cells act as both as the immune cells in the brain to protect the brain from the infection and also to prune away the unnecessary synapses during development?
Yeah, so by and large, microglia are pretty similar throughout the brain. There are some subtle differences, but what happens is that microglia are going to change depending on what's happening in the environment. So if there's an infection, microglia will then adopt a state that is responding to the infection. If there's damage, the microglia that's next to the site of damage is going to change and respond to that. After it's resolved, it will then go back to being sort of a normal looking microglia. So within the brain, there will be different microglia states depending on what's happening within the local environment of that one microglia.
Got it. Thank you. We have another question. Lisa is asking, how does this possibly relate to treatment of neurological disorders, for example, epilepsy?
Yes. So this would be important for neurological disorders in which you have loss of synapses. That said, microglia have lots of other roles in the brain beyond responding to infection and removing synapses. Some people have shown that they can release molecules that affect the level of activity within the brain, which might be something that's important within epilepsy. But in terms of treating disorders where they have removal of synapses, if we are able to identify the way that microglia determine which synapses to remove or the signaling between microglia and astrocytes, we might be able to look at that as a potential way to prevent the synapses from being removed. Whether that is good for the disease or bad for the disease, I think that might be something that comes up later in the second half of this, but this is important for these disorders.
Thank you. We have another question about synapses. Does the synapses pruning also exist in other animals, especially in invertebrates?
Right. So in every, I would say every system where you have synapses, there is going to be plasticity in terms of synapses that get formed and synapses that get removed. Microglia, being immune cells, are not present in invertebrates, but there are similar cells that play a similar role in invertebrates. So astrocytes, which I mentioned in vertebrates, have an important role in sort of shielding the synapses from the microglia. They also have the ability themselves to remove synapses, and in invertebrates, they have been shown to, in certain cases, remove synapses themselves. So a lot of the rules and general principles are true, but the specifics of which specific molecules and which specific cells varies between species.
Nice, thank you for that. Sean is asking, once synapses are lost, can they be restored somehow? Do the mice regrow whiskers? And if so, does the region of the brain become active again?
Right. So your brain is forming new synapses all the time. You may not form exactly the same synapse, and it may have, you have multiple synapses between pairs of neurons. So you may lose some, but you would still retain others, and you might form new ones. And this will all affect how activity, travels through the network. To the question about the whiskers, generally, if they grow back, then you would expect activity to return, and then you would expect not as many synapses to be removed because the amount of activity wouldn't go down as much. What we try and do to prevent that is we use a if you've done like to basically damage the follicle, and so that the whiskers won't grow back. And that way we can make sure that we don't have to keep cutting the whiskers, because every time you cut them, you probably move them a bunch. And then that is a sense of touch, which creates more activity when you really want less activity.
Thank you. Another question is, how does this rate depend on the age of the mice? Is there an age where pruning is not observed anymore?
Yeah, this is actually one of the projects that we have ongoing in our lab right now. We've made the observation, if you remove the whiskers from the mouse too early, you don't ever form this map where you have this part of the brain responding to one whisker and this other part responding to the other. If you remove them too early, you never form that. And people call this a critical window. If we wait until after that window, there is actually a different window for a different process, which is after the map is formed. If we remove the whiskers, we normally see this removal of the synapses and preservation of the map. But if you wait until adulthood, when you remove the whiskers, the synapses don't go away. And what's interesting is that astrocytes develop over the course of this period, and they form more and more contacts with synapses as they develop. And we're actively testing this idea that what if we make the astrocytes contact less synapses? Can we make the brain more plastic in adult? What if we make them contact more synapses in development? Can we make the brain less plastic earlier on? And this is something that's very important within sort of neurodevelopmental disorders, where it's thought that there are changes in the timing and regulation of these critical windows of plasticity.
Nice, thank you. Jill is asking, are the microglia recruiting other molecules to help with their different functions?
Yes, so a microglia would be using different molecules for different functions. I mean, they're regulating neuronal activity. They would be releasing molecules that tell the neurons to increase or decrease activity. If they're communicating with astrocytes, they would use molecules that tell the astrocytes whether to move away from the synapse or not. And what's interesting is that neurons and astrocytes can both sense both types of signals, but because they are different types of cells, they would each respond in their own specialized way.
Thank you. Allison is asking, I know you mentioned autism, but are there any studies that show any activity with dementia or Alzheimer's individuals?
Yes, so I think Pat is giving a talk a little bit more about the second, stage of life, and what happens to synapses when you already have them and you want to keep them. But if you have reduced activity in a synapse, it would still get removed.
Thank you. Liz is asking, are there any conditions in which microglia can become overactive? If so, what are the repercussions, for example, in excessive pruning?
Yeah, there are definitely many instances in which microglia can be put into a state in which they are removing more synapses than they normally would. What we're doing in the mice, by removing all of the whiskers, we remove way more synapses than we normally would because we've reduced activity. You could also think about an instance where microglia are releasing more of that molecule that signals to astrocytes to move away. That might cause more synapses to be removed than normal, or something happening within the astrocytes, irrespective of any signal from the microglia, that causes them to contact fewer synapses. And in all of those cases, you would end up with more removal of synapses and fewer connections in the brain than you would typically.
Thank you. And our last question is asked by Naman. He is saying that the graph that you showed from the Nature publication showed, or the Nature reviews showed a lower synaptic pruning or higher synaptic density in autism spectrum disorder. And he is curious if this is due to reduced function of microglia or if there are other factors at play.
Right. So within the brain, there are many different parts of the brain, and each one of them is going to be a little bit different in terms of how many synapses gets removed, what time of development they get removed. And I've sort of summarized, broadly speaking, in the cortex, you have more synapses, and you see them more synapses in a patient with autism than in a neurotypical individual. This is between the balance of these two forces I mentioned. One is the formation of new synapses, and the other is between the removal of synapses. And so it's probably both of these things in combination that leads to more synapses in a patient with autism spectrum disorder. And there are a lot of different genetic factors and environmental factors that have been shown to play a role in influencing both of these two aspects of the number of synapses you have in the brain.
All right, thank you. I don't see any more questions, so I think we can proceed to the next speaker. Thank you again. And if anyone else has questions, feel free to type them in the chat, and maybe Travis can follow up in the chat. Thank you.
Thank you, Travis. It was very interesting and we learned a lot. With that, I would like to invite our second speaker, Patrick Sheehan. He's a postdoctoral associate in the lab of Dr. Dorothy Sheffer at UMass Chan Medical School. He grew up around Gettysburg, Pennsylvania, and attended Clarion University there for his undergraduate work. Patrick graduated with a degree in molecular biology and then spent a year at NIH researching on malaria. He did his PhD at Washington University in St. Louis, studying circadian rhythm in Alzheimer and Parkinson disease. He joined Dr. Schiffer's lab at UMass Chan in 2022, and now he's studying how synapses are lost in neurodegenerative diseases. And outside of the lab, he likes to hike, cook, and play frisbee. So with that, I would like to welcome Patrick.
Okay. Hi, everyone. Can you all see my laser pointer and screen okay? Okay, perfect. Okay, so I want to thank the UMass Postdoc Association for having me. And I want to thank all of you for showing up. I also want to thank Travis for doing such a nice job introducing a lot of neuroscience topics and making my job a lot easier. So kind of similar to how Travis talked about how synapses are lost in development and how that's normal, I'm going to be talking about how synapses are lost during neurodegeneration and how that can result in symptoms. So unfortunately, many of us are intimately familiar with at least one type of neurodegeneration. It's estimated that about 50 million people worldwide have a neurodegenerative condition. And there are many different types of neurodegeneration, but four of the big ones are Alzheimer's disease, Parkinson's disease, multiple sclerosis, and Huntington's. And of course, there's others like ALS. And many of these neurodegenerative conditions have their own symptoms. So, for example, people with Alzheimer's disease generally have difficulty with memory, difficulty with critical reasoning or coordination, and many times they have behavior or switches in their mood. Whereas people with multiple sclerosis, they also have mood changes and difficulty with cognition, but something that's more unique to MS would be numbness and pain in their extremities, or muscle weakness and loss of vision. So there are many risk factors for developing a neurodegenerative disease. Of course, there are environmental factors and genetic factors that play a role. But by and large, the number one risk factor for developing a neurodegenerative disease is aging. So you can see by these graphs, on the x-axis is people's age, increasing age over time. And on the y-axis is the cases per 1,000 people or cases per 100,000 people. And you can see pretty simply that as you age, the number of cases of both Alzheimer's and Parkinson's goes up. And you can also see by these graphs another risk factor for neurodegeneration, which is sex. So women tend to get Alzheimer's disease a little bit more, and men tend to get Parkinson's disease a little bit more. There are a number of hallmarks of neurodegeneration that are fairly common across most of these diseases at a fairly cellular and molecular level. Many of us are familiar with neuronal cell death, which is kind of the overall cause of neurodegeneration. And many of us are also familiar with pathological protein aggregation, for example, something like amyloid beta and tau, which aggregates in Alzheimer's disease. But the thing I want to focus on today is this one, synaptic and neuronal network defects. And the reason I want to focus on this is because synapses correlate fairly closely with symptoms during disease. So this is a graph of cognition of patients with Alzheimer's disease. And you can see as the person ages and the disease progresses, their cognition goes down to eventually they hit a dementia phase. And then on this orange line here, you can see the amount of synapse loss. So as the disease progresses, you get more synapse loss, which results in less cognition. So the two are very closely related, where as you get more synapse loss, you get less cognition and more symptoms. So we're really interested in how these synapses are lost during disease. Travis talked a little bit about how these synapses are lost during development. And during disease, it can follow a slightly different mechanism. So this is just a normal synapse. And over the course of neurodegenerative disease, there are these proteins that are expressed and land on synapses that we like to call eat-me signals. So C3 here is an eat-me signal that we like to study. And these eat-me signals signal to microglia to come and eat that synapse. So eventually that microglia recognizes that eat-me signal that's on the synapse, and then the microglia will eat the synapse, resulting in synapse loss. So as synapses are going away in the brain, one of the reasons that they're going away is because microglia are eating them. So our lab has been studying this process for quite a while. And one of the ways that we study this process is using a mouse model of multiple sclerosis. This is also called experimental autoimmune encephalomyelitis, or EAE. You'll see in some of my pictures and later slides. But just remember that this is a model of MS. So this is really basically how it works. We take normal mice, we inject them with a protein to cause an autoimmune response. And then over the course of a few weeks, these mice get limb paralysis and they get neurodegeneration in a fairly similar way as MS patients do in the clinic. So we can use this model to study how synapses are lost during disease. So just to reorient you again on the structure of a neuron, this is what a typical neuron might look like. Synapses are at one side of the neuron, and on the other side is the cell body or the neuronal nuclei. And we can image these on a microscope and look at these during over the course of disease. So if we look at our synapses during our EAE or in our mouse model of MS, we can see that synapses are lost during disease. You can see much less of this white staining during EAE, suggesting that these synapses are going away. And then interestingly, if you look at this stain for neuronal nuclei, you can see that the number of neurons are staying the same, suggesting that degeneration is going in this direction, where synapses are lost first before the neuron cell body goes away. And that's exciting to us because it might suggest that if you rescue synapses, you might also rescue later neurodegeneration. Okay, so how are these synapses being removed or going away over the course of this of MS? So I want to bring up what we talked about a little bit earlier, these eat me signals. Again, one of these that we like to study is called C3. So once again, we can stain synapses in our mice, which are labeled green here. And you can see that there's less green synapses in our MS model. And then if we stain for C3, which is this red color, you can see much more C3 in this in our MS mouse model, suggesting that this eat me signal is being highly upregulated. And if you can see, this red signal overlaps with the green signal, creating these yellow dots, suggesting that this eat me signal is landing on neurons or landing on synapses. Now, how are these, what's happening after these synapses are tagged with C3 or this eat me signal? We can look at microglia, which we stain with green here. And then we can also measure the number of engulfed synapses inside of these microglia. And we can see in our control microglia, these microglia rarely eat synapses. But in microglia in an MS model, you can see a number of synapses inside of microglia, suggesting that these eat me signals are in fact landing on top of synapses and causing microglia to come and eat them. So to make a really long story short, this is kind of the model that we work with in the lab, where over the course of disease in multiple sclerosis and in other neurodegenerative conditions, these eat me signals land on synapses and cause microglia to come and eat those synapses, and results in synapse loss and results in downstream pathology. But our lab has found that if we express this protein called curry, which is a C3 inhibitor, on the synapse, we can prevent these eat me signals from binding, and we can rescue synapse loss. So we think this is really exciting. It's a really novel way of preventing synapse loss during disease. And luckily, we're not the only ones to think this is interesting. Companies are currently running clinical trials, like this company called Annexon. This is just a screenshot of a news snippet of this company Annexon running clinical trials on patients with Huntington's disease, targeting these eat me proteins like we have here. So we're hoping this is kind of generally applicable to other forms of neurodegeneration, and we're hoping that targeting these eat me signals and preventing microglia from eating these synapses will help delay symptoms in many of these diseases. So with that, I want to thank everybody. Again, me and Travis are both in Dory Schaefer's lab here at UMass, and there are a number of other people in our lab that we work with that are fantastic and have helped a lot with this work. And with that, I will take any questions.
Thank you, Pat. That's very interesting and very exciting. So if you have, for the audience, if you have any questions, please type in the chat box. And while you're chatting, we have one question from Allison. The question is, do the synapses and microglia cells affect the axon nerve cells?
Do the synapses affect the axon nerve cells? So the microglia. So what I talked about in the talk is microglia come and eat the synapses that are on the neurons. So they affect neurons in many other ways. Travis briefly touched on it. They can signal to neurons to send out chemical messages more or less. But yeah, they have a very close connection, and they talk to each other all the time.
And also, Mina has a question. Where does the eat me signal get secreted from? Does it come from the neuron, and who decides which synapses has to go?
Oh, a couple of good questions, Mia. Okay, so where does the EV signal get secreted from? That's a project that we're working on right now. We think it's from a different glia called astrocytes that Travis talked about a little bit. They seem to secrete a lot of this C3 eat me signal, but that is yet to be determined. So no, it doesn't come from neurons. And then who decides which synapse has to go? So we're not positive what's causing these proteins to land on synapses, but it seems like around places of pathology where there's a lot of inflammation around amyloid beta plaques or other places where there's a lot of inflammation in the brain, there's upregulation of these proteins. So they probably land on neurons that are kind of close to these pathological proteins. So microglia are trying to clear them and get them away. But it's also really likely that these proteins are landing on nearby neurons and causing the loss of synapses in relatively healthy neurons as well. So I assume it's not super specific, and it's just a mechanism downstream of inflammation.
Okay, thank you. So Alessia asked a side-by-side question, and also Lisa asked like a kind of following up questions. It's very logical questions. So the first part of Alessia, you already answered it, is which cells makes these eat me signal. But Alessia's second question is, can those cells which release these eat me signals be blocked from releasing it or making it in the first place? And following this question, Lisa also asked a question, will this research lead to new drug developments to treat neurological disorders?
That's the goal. That's the goal, Lisa. So, Lisa, can these cells be blocked from releasing it? I think that's what current drug development is trying to do. It's a little bit difficult because these eat me signals actually have really important roles in the peripheral immune system outside of the brain. So any drug that you give should be directed at the CNS. So a lot of these drugs that they're making, they're injecting into the CSF, which I think is the right way to do it. So yeah, I think can the cells be blocked from releasing it? I think so, and I think that's where some of the drug development's going to prevent synapse loss.
Okay. And Liz asked another question from another angle. Does the C3, the eat me signal, have any positive functions that we need to preserve, which would make us not to want to prevent it from being released entirely?
Yeah, really good question. So I touched on it briefly, but in the peripheral immune system, outside of the brain, in your liver and spleen, C3 has some really important roles in general immunity and protecting your body from infection. But that's not really a worry in the brain because the brain is immune privileged for the most part. So we don't get infection there a lot. But there is projects in the lab looking to see if there are any other functions of C3 other than tagging synapses for phagocytosis or to be eaten. We're not sure, but we're hoping that even if it does have different functions, that we can target those different functions of C3 in different ways and still rescue synapse loss while retaining maybe other positive parts of C3.
And Alessia has another question. Are there any other immune cells which are involved in eating synapses or just microglia?
Yeah, good question. So microglia have been studied by far the most. I think they were the obvious choice. They are the kind of canonical immune cell in the brain. When you think of a cell eating something in the brain, you think of microglia. But there's been a lot of recent research on other glia cell types, some that Travis talked on, like astrocytes and certain oligodendrocytes that can also eat synapses in certain situations. So if microglia aren't able to eat synapses, there's a chance that they can signal to another one of these glia cells to come and eat that synapse for them.
And I also have a question for myself. So when you use the curry or whatever the inhibitor of C3, do you see any pathology or pathogenic effect other than the cure?
We didn't notice any negative effects. If that's what you're asking. So in this model, in this MS model in mice, vision decreases over time. So when we give curry, we rescue synapses and we rescue vision loss, and that was our main output for this for that assay.
Okay. Right. So if there's no more questions, thank you for your exciting talks and Ruchi.
Thank you, Patrick. It was really informative, and we enjoyed your presentation. And with that, I would like to thank our speakers, Travis and Patrick, once again, and to everyone who has joined and asked questions and be the part of discussion. And we also, please, we would like to ask you to fill one survey form, which will be in the chat box soon. And you can take a moment so that we can get your feedback and make these events even better. Thank you very much for your time.
Thank you.
Have a good evening. Yeah.